My name is Elizabeth Papish. I'm a chemistry professor at the University of Alabama. My group does catalysis, and we're at the intersection of organic and inorganic catalysis. I'm really interested in using catalysts and making catalysts to store energy in fuels, preferably liquid fuels, since that's convenient for transportation. We also have an anti-cancer project where we're making ruthenium compounds for anti-cancer properties. We believe that they can be selectively toxic towards cancer cells. A student of mine came down with cancer, so we started reading and thinking a lot more about it and reading more papers on it. And we realized that one of the compounds that we were making actually looked a lot like an anti-cancer compound that was in the literature. So we've been exploring that and making analogs that are pH sensitive to hopefully take advantage of the low pH in cancer cells.
Hi, thank you for being with me today. I'm here to tell you about the work my group does. Thank you for watching that short video about my group. I'm going to tell you today about our work on anti-cancer and catalysis. The anti-cancer project has NIH funding, and then the catalysis project has NSF funding. And I'm going to tell you all about our work. And I just also want to emphasize right off the bat that UA is a friendly place. We're here to help you. We're here to help you succeed. We can bring you up to speed if there's things you don't know. We try to work closely with you and give you all the tools you need to succeed. And now I'm going to start my presentation and tell you about the work that my group does. The project has NIH funding, and we would love to get 1 or 2 students involved in that project. We're working on protic and light-activated ruthenium anti-cancer compounds. And there are a lot of different ruthenium anti-cancer compounds in the literature. There are a lot of them that use nitrogen ligands. But what's unique about our work is the fact that these hydroxy groups are here that are pH sensitive. So we're able to change the structure as a function of pH. That's interesting because of the pH effects in cancer cells, where sometimes cancer has a lower pH. And we see here that the hydroxy groups are converted to O- groups in cellular media around pH 7 to 7.5. And so that's a very interesting and fundamental change, that by removing those 2 protons, we're changing the structure. And we're able to look at a fundamental study of how does that change in structure affect the properties. It turns out that this form that we have in the cancer cells has better uptake, and it's also very effective at converting triplet oxygen into singlet oxygen, and singlet oxygen is a toxic compound. This leads to the good toxicity that we observe against cancer cells. And we can also say that this is very interesting because this uses the FDA-approved technique called photodynamic therapy, where you can shine light on the cancer cells and you can imagine getting the toxicity effects in the cancer cells and not getting any killing effect in the normal cells, because the light can be specifically targeted for the cancer cells. So we think that's very interesting. Following a series of papers my group published on these types of compounds, we have recently been working on a follow-up paper using these compounds with these bathophenanthroline ligands, improving the cellular uptake. And this allows us to get an IC50 value of 0.5 micromolar. Recall that the IC50 value is the concentration that kills half of the cells. This is an excellent value in the nanomolar range at 500 nanomolar. We also look at the ratio of dark toxicity to light toxicity, and ideally we want a value of hundreds or thousands, and so this is an excellent compound at around 2,000. This has been the work of several students in my group and in Dr. Kim's group. Dr. Kim at UA does all our cellular studies, whereas we do the synthesis and the spectroscopy studies. So again, everything that we do in the group varies from synthesis, crystallography, UV vis. We measure how well the compounds would be taken up into cells using octanol and water as a model. We do photochemistry and computations with collaborators. And then with the Kim group, we do biological and medicinal studies and measure uptake in cells and look at how the compounds are distributed in the cells. We've published several papers on these compounds in the last few years, including this one that we recently submitted in 2020. We do everything from studying the fundamental chemistry of the compounds to studying the biology of the uptake and how they distribute in cells. And this recent paper was on the photochemistry of these compounds. Switching gears for a moment, my group also does catalysis studies, and we have an NSF-funded project on that topic. Just a little bit of background on catalysis. It's speeding up reactions. It's how reactions are typically done in industry, where you need a catalyst to speed up the reaction. Every time you go around the cycle, that's one turnover. And an effective industrial catalyst is going to do this reaction millions of times. Like the way enzymes do, they may go around the cycle millions of times before the enzyme is no longer effective. And we're hoping to do the same thing with industrial catalysts. We're looking at methods for taking carbon dioxide, an abundant greenhouse gas, and using it to make carbon monoxide, because that gets us to fuels and fuel precursors. We can also make bulk chemicals with that. So this is a solution to the energy problem because it can allow us to make liquid fuels and also a source of synthesis building blocks in terms of the carbon monoxide. So again, it's an energy related problem. We need to figure out how to store energy and obtain energy as petroleum reserves get depleted. In this project, we're also doing a lot of organic and inorganic synthesis, including making organic ligands and looking at the pH effects of these ligands, looking at how the ligands change with removing protons and also with other substituents like methoxy groups, also changing these rings and looking at substituent effects here, and trying to understand how to make better catalysts. On this project as well, we've published a number of papers on nickel catalysts that switch on or switch off with protons, and also looking at ruthenium catalysts. The key thing is that the substituents and the catalyst structure plays a key role in all these papers. The only problem with these papers is that we're using a photosensitizer here, using iridium as a photosensitizer, which is a precious metal and a very expensive metal. We'd like to eliminate that. And that's what many of these students have been working on. Sanjeet has been able to eliminate the photosensitizer and use a ruthenium catalyst shown here, where these aromatic rings help to increase the light absorption and allow this catalyst to be both a photosensitizer and a catalyst. We're also able to get around 33,000 turnovers. Remember we talked earlier about how many times you complete that cycle, how many times the catalyst does the reaction before decomposing. And this compound can do it 33,000 times before it decomposes. And this one is record setting because the compounds that are both photosensitizers and catalysts have been quite rare in the literature. Other new directions my group is going in include using first row metals using a low valent strategy. We're looking at cobalt, nickel, and iron catalysts. We're looking at a coordination sphere restriction strategy, trying to block off sections of the access to the metal using steric bulk and getting metal catalysts using iron and cobalt that way. We're looking at photosensitizer catalyst assemblies and looking at surface attached catalysts. These are all methods for getting highly active catalysts that can function with, let's say, the source of electrons being water oxidation, for example. So these are all parts of a green energy solution, how you would get energy once you run out of fossil fuels, for example. Again, in this project, we're using synthesis, making new ligands, new functional groups, new metals. We're doing crystallography and spectroscopy. When we're doing catalysis, we're looking at measuring what occurs and measuring what the products are and how many turnovers. The students on this group have been mostly organic and inorganic students. All of these students have worked on catalysis, and these students on the left have worked on anti-cancer. Again, in addition to organic and inorganic, we also do a lot of synthesis, catalysis, mechanistic studies. Lastly, I'd like to say that a PhD takes several years. You should do what you love. And these students have published 3 to 5 papers and have gotten jobs in academia and industry. If you get the chance to visit us, come take a look and see the instruments that we have in our group. And we have a number of instruments for synthesis and for spectroscopy. I invite you to meet with me on Zoom or come visit in person if you get the opportunity. And lastly, I'd like to thank NSF and NIH and all my collaborators. And a lot of students have really worked on this project and paved the way on this project. If you come to UA, we're here to help you. We're here to help you succeed. And we really do hope to help you learn what you need to get a fantastic job and be successful. Thank you.